Hi, welcome to Exam Prep Week for the 2020 AP exams. Today we're going to debrief the scoring guidelines for practice exam number 2. It had 2 questions. Hopefully you guys were here yesterday and you have your responses because we are going to go over the scoring guidelines for both of those. So if you have them, grab them. Question 1 was interpreting and evaluating experimental results. You had 25 minutes to read the question and write a response with an additional 5 minutes to upload. This question is a 10-point question with 10 task verbs and will compose 65% of the total exam score. Question 2 is on conceptual analysis, and you had 15 minutes to read the question and write your response with an additional 5 minutes to upload your response. This is a 6-point question with 6 task verbs and will make up 35% of your total exam score. So I would like to welcome our very special guest today. It's Dr. Amy Dykstra, the chief reader for AP Biology. So welcome, Amy.
Hello, it is very nice to be here with you. As Ms. Kate said, I'm Amy Dykstra. I'm a professor at Bethel University in St. Paul, Minnesota. I've learned how to say Minnesota since I've lived in Minnesota. And I have been involved with AP Biology ever since I was an AP Biology teacher, starting in about 1994, and I've been reading AP Biology exams and scoring AP Biology exams since about the year 2000. And so I'm delighted to join you and talk a little bit about the scoring guidelines for this long question. So let's move to the question. So we're just going to read through it again to refamiliarize us with the question. This is, again, the long question. Electrons from the oxidation of glucose and other nutrients by glycolysis and the Krebs cycle are added to the electron-carrying compound NAD+ to form NADH. To meet the continuous requirement for NAD+ by cells, NADH is reduced back to NAD+ through the electron transport chain in the presence of oxygen, or through fermentation in the absence of oxygen, as shown there in Figure 1. And then we have this Figure 1, which is a simplified model of metabolism and the recycling of NAD+. MTND5 is a mitochondrial gene that encodes a subunit of NADH dehydrogenase, the enzyme that catalyzes the initial oxidation of its substrate, NADH, to NAD+ and H+ in the electron transport chain of mitochondria. A mutation in MTND5 is associated with a rare genetic disorder that results in a buildup of lactic acid in the body. A researcher hypothesizes that the mutated NADH dehydrogenase has decreased activity but is not completely non-functioning, and that by increasing the pool of NADH in cells, the activity of NADH dehydrogenase will increase. To test this idea, the researcher treated a group of individuals with this disorder with a vitamin that is similar to NADH and measured the concentration of NAD+ and lactic acid in the blood over the course of 20 weeks. The results from a representative individual are shown in Figure 2. So let's look at the graph. This is Figure 2, and so when you look at these graphs, you want to notice what's on the x-axis and what's on the y-axis. So time is on the x-axis, and the top graph is looking at the concentration of NAD+, which, as you can see, goes up for the first 5 weeks, and then it starts declining for the rest of the experimental time. And then the concentration of lactic acid goes down for the first 5 weeks and then kind of levels off, goes down a little bit. So let's go on to the scoring guidelines, or actually to the actual questions. So notice that there are 10 questions, A through J. The format of this exam looks a little different than you may have been expected. You may have been expecting paragraphs, And I would just remind you that this exam this year is a special one-time only version of the AP Biology exam. So the formatting is a little bit different, but each question, each task that we are going to ask you to do is going to be associated with a letter. So you'll notice that there is a bolded task verb with each A through G. So let's go through the questions. A is describe the pattern of inheritance that is most likely associated with the mutation in the MTND5 gene. B, explain why individuals are not typically heterozygous with respect to mitochondrial genes. C, identify a dependent variable measured in the researcher's experiment. D, identify one control that the researcher could use to improve the validity of the experiment. E, justify the researcher analyzing blood samples at many intermediate time points instead of at only the beginning and the end of the 20-week period. F, describe the relationship between the concentration of NAD+ in the blood and the concentration of lactic acid in the blood during the first 5 weeks of treatment with the vitamin. G, based on figure 2, calculate the average rate of change of blood NAD+ concentrations from week 5 to week 17. H, the researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells, predict the effect of the MTND5 mutation on the rate of oxygen consumption in muscle and brain cells. I, justify your prediction. And finally, J, the researcher had hypothesized that the addition of the vitamin that is similar in structure to NADH would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Explain how the vitamin most likely increased the activity of the enzyme. So I hope you have your answers ready that you wrote when you took the practice exam. And so let's look at the scoring guidelines and let's kind of walk through them. So letter A, describe the pattern of inheritance that is most likely associated with a mutation in the MTND5 gene. And it looks like Ms. Kate has put up some figures here that you may remember from her lectures earlier. And this is to remind you that a mitochondrial gene is found on a single chromosome and that it's only inherited from the mother. So the correct answer, the scoring guideline is that the mutant allele is only inherited from the maternal parent. Another way you could say this is that the mother passes the gene to all of her children. So that's part A. And notice that when there is a describe, usually we are looking for more than one thing. So we are saying that it's inherited only from the maternal parent, or the maternal parent passes it on to all of her offspring. So that is a describe what's happening. Part B is an explain. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. And if you look up in that top left image there, we see a circular chromosome like what would be found in the mitochondrion. And so the answer, the acceptable answer, is that MTND5 is a mitochondrial gene, and mitochondria only carry a single unpaired chromosome. Another way I thought about this question is that in order to be heterozygous, you have to have two different alleles, one coming from the father and one coming from the mother. And so you're only getting that mitochondrial gene from the mother. So there's no way to be heterozygous. And you can see in that other diagram how how there are paired chromosomes in nuclear inheritance, but mitochondria only have the one chromosome. So there's no pairing of the chromosomes. Yeah, okay, so that's letter B. So when you see an explain why there, oftentimes you might want to think about writing a because. So explain why, well, it's because what? So think about that with an explain why, and explain how would be asking you more to lay out how something happens, but an explain why, think about a because. Okay, then letter C is an identify question, which means that we're only looking for one thing with an identify, whereas describe or explain, we're looking for a little bit more of an answer. With an identify, we're looking out for one thing. So identify a dependent variable measured in the researcher's experiment. So remember, if you look at on the left there, Ms. Kadea said the dependent variable is what you are measuring in your experiment. It is what is on the y-axis. That's the dependent variable. So what is measured? Maybe, Ms. Kadea, if we could skip back to the graph so we can see this a little bit bigger. You can see what's on the y-axis there is concentration of NAD plus for the top graph, or concentration of lactic acid. So we would accept either one of those answers. You would only have to give one because we've only asked for one. So concentration of lactic acid in the blood or concentration of NAD plus in the blood. Ideally, you would write this out as a complete sentence, but we are just looking for that one thing. Okay, letter D, again, is an identify. So we're just looking for one thing to identify. Identify one control that the researcher could use to improve the validity of the experiment. So an experiment should always have a control. The experiment that was described in our question, they just talked about measuring blood in one person. To really know how your results compare, you really need a control. And so the appropriate control is to measure NAD plus and lactic acid levels in a group of other affected individuals, so other people with the same mutated dehydrogenase gene, but these people would be treated with placebo. Or possibly we might accept if you said not treated with the fake vitamin or the vitamin that's like the NADH. Really the best control is a placebo. So you're giving somebody a pill every day, just like the vitamin taking people are getting the pill every day. But that pill, that placebo, is just a sugar pill. It doesn't have that vitamin in it. So that would be the appropriate control. Letter E, justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and the end of the 20-week period. And so the answer to collecting more data points is that you're going to have more data. And so this will give you a more accurate trend. It gets you more of a picture of what's happening over the entire course of that experiment. Likely the researchers didn't know how long it was going to take these patients to respond to the treatment with the vitamin. And so they wanted to take time points all along, over several time periods. They wanted to take samples, and this just gives them a more accurate picture of that entire trend of the whole experiment. All right, so that's the first half. So now the second half, F through J. So let's look at F. Describe the relationship between the concentration of NAD plus in the blood and the concentration of lactic acid in the blood. So we're asking you to describe how these two things are related. So you can say that the concentration of NAD plus increased and the concentration of lactic acid decreased. That's probably the simplest way to say this. You could say that as the NAD plus increased, the lactic acid decreased. And remember, this is just the first 5 weeks of the treatment. Ms. Kate, let's back up to the graph again one more time so we can see that graph a little bit better. Notice that it's just the first 5 weeks of the experiment, and that was the time period over which that concentration of NAD plus was going up. And then at the same time period, if you look on the bottom graph, you can see that the lactic acid was going down. Some of you might have expressed this as an inverse relationship, and that would be okay as well. That would also be acceptable. So let's go to the next one. Letter G. Based on figure 2, calculate the average rate of change in blood NAD plus concentrations from week 5 to week 17. And so notice that we are looking for a rate, and in your equations, rate, you can find the rate equation is dy dt, d meaning change. So the rate is always a change in y over the change in time. I always think of a rate as being a slope of a line. If you think about that in math, and I always learned it as rise over run. How much does it rise over how much it runs? So that's how I always think about it. And so if we look at the graph, and I think you can kind of visualize the graph. Well, yeah, this is great. So we're asked from week 5 to week 17. Be careful that you don't go all the way to week 20. Actually, when I was going through these questions, that's what I did. I got the wrong answer. Even the chief reader can make mistakes, but not very often. Okay, so we're going to take the final amount and subtract the initial amount. So we want to take 60 as our final concentration of NAD plus at week 17, and we're going to subtract 300. So that should give us a negative number. And then from week 5 to 17, you should be able to do that math in your head. That should be a difference of 12, 17 minus 5. So let's look at the equation again in the answer. So we have, and the important thing here, guys, is that you set up the question correctly. That's what we're going to be looking for, not necessarily the answer. You can give us the answer, but we're especially going to be looking at the setup of the question. So make sure you show your work. 60 minus 300, as we said, that's minus 240 over 17 minus 5. So minus 240 over 12. Most of you could probably do that in your head to get a minus 20. I think minus 20 really is the best answer here because it is going down. So it's the change, it's a negative slope. It's going down. But notice that the important thing is to have the correct setup. Okay, so that's letter G. Letter H. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the MTND5 mutation on the rate of oxygen consumption in muscle and brain cells. Now, when we ask you to make a prediction, you just have to give us your prediction. You do not have to explain your prediction for that task verb, for the task verb predict. You just have to give us your prediction. In this case, the correct prediction is that there would be a decrease in oxygen consumption. Now notice that we do follow up this prediction by asking you to justify your prediction. And this is where you will explain how you figured out how to make your prediction. So if you think about it, this mutation actually prevents the NADH from donating its electron to the beginning of the electron transport chain. So if there aren't any electrons going into that electron transport chain, then there isn't going to be a need for an oxygen at the end of that chain to receive that electron. So remember that the oxygen is the final electron acceptor in the electron transport chain. That's the whole reason that we need to breathe, is to have oxygen for our electron transport chain. And so as the wording is here in letter I, the mutation likely inhibits the rate of oxygen consumption. You've already said that in your prediction. Here's the because, and this is the justify: because the electron transport chain will be less efficient. This is because there's fewer electrons going into it, because we can't dehydrogenase, we can't dehydrogen. The dehydrogenase can't do its job of stripping the hydrogen off and donating that electron to the electron transport chain. I'm not sure what the verb is there. Dehydrogenate, maybe, I don't know. I'm not sure that's right. But anyway, the mutation is going to decrease the ability of that enzyme to do that job. So therefore, less oxygen will be required as the terminal electron acceptor. Hopefully you followed my convoluted explanation there. Okay, so let's move on to J, our last question. And in this one, the researcher had hypothesized that the addition of the vitamin that is similar in structure to NADH would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Here's the question: explain how the vitamin most likely increased the activity of the enzyme. Well, we told you that the enzyme is similar in structure to the NADH. So that suggests that the vitamin is going to act just like the NADH. So the answer we're looking for here is that the enzyme had decreased activity but still functioned. This is kind of part of what the researcher thinks is going to happen. Increasing the amount of substrate should increase the amount of product produced because, and here's your explain, here's your because the vitamin was similar to NADH, it could bind to the active site. That is where that molecular similarity comes into play. It binds to the active site of the enzyme and effectively increases the substrate concentration, i.e. apparently that vitamin is acting like an electron donor and a hydrogen gets pulled off of it, just like what would happen with NADH. So, so we think that so we've got a picture of a competitive inhibitor, but you could kind of sort of think in your mind about crossing out competitive inhibitor and replacing that with vitamin. And the NADH is kind of the natural substrate there in yellow. And so what's happening is the vitamin is actually going to bind to that active site, and the enzyme is going to do its action. It's going to do its enzymatic activity on that vitamin rather than on NADH. So there are the scoring guidelines. And so I now have a chance to just give you some words of wisdom. And so one thing I would say is that you may not be able to answer an entire long question. We have kind of intentionally given you a limited amount of time to work. So if you see part of the question that you're not sure how to answer, it's okay to skip and go on to the next question. And if you have time, write an answer to everything. If you have, yeah, but don't get too stressed out if you can't answer everything. And there are a lot of clues in the question. Try to just remember that we are trying to test your biology knowledge. Even if you've never heard of NADH dehydrogenase, you do know about glycolysis, you do know about fermentation, you do know about the electron transport chain. So apply what you do know to what you read in the question, and we've tried to write questions that will ask you, that will that will pull your biology knowledge out of you. So, so we are excited to see what you're going to show us. And yeah, that's my main words of wisdom. Try to address the questions with what you know about biology.
Awesome. We thank you so much for joining us, Professor Dykstra, and we look forward to seeing those questions on May 18th. I'm going to go over question 2 now. So question 2 shows you figure 1, which is a phototropism experiment. And you can see that there are 4 treatment groups, and you're going to be making a prediction about treatment group number 5. The question reads, phototropism in plants is a response in which a plant shoot grows toward a light source. The results of 4 different experimental treatments from classic investigations of phototropism are shown in figure 1. You will need to make a prediction about the last treatment group. This has parts A through F, and again, you can see that the task verbs are bolded for you. So in part A, you're asked to identify the treatment group that represents the control in this experiment. In part B, you're asked to explain how phototropism is an example of a cell signal pathway. In part C, you're asked to look at treatment group 4, and in treatment group 4, the tip of the plant is removed and placed back onto the shoot using a permeable barrier. You're asked to explain the effect of this change on the response seen in treatment group 4. In part D, you're asked to justify the claim that the cells located in the tip of the plant shoot detect the light. Use evidence from the results from treatment group 1 and the results from treatment groups 2 and 3 in your response. In part E, a 5th treatment group is added that has the tip of the plant removed and placed back onto the shoot using an impermeable barrier. Predict the response of treatment group 5. And then in part F, you're asked to justify the prediction you made in part E. So let's take these one at a time. Each one of these is earns 1 point for a total of 6 points on this question. So identify, remember as Professor Dykstra told us, you just need to identify. We're looking for one thing, and in this case, we want to identify the group that represents the control in this experiment. And treatment group 1 is identified as the control, and this is because it has an unaltered tip. It can be used to compare to the other treatment groups with altered tips. So you could just say treatment group 1 is the control group, and that would earn the point. But there's a little bit more information about what makes it the control group, and you know, because you have good experimental design knowledge, that the control is what we use to compare the other treatment groups with. Part B, explain how phototropism is an example of a cell signal pathway. And you might recall from some of our lessons that there are those 3 basic stages. There's reception, transduction, and response in a cell signal pathway. So in this case, the signal is the light. Photoreceptors in the plant tip receive the signal, and we call this reception. There's some shape change at the reception of the receptor, and then the tip produces a substance in response to the light that affects the plant stem. And so there is a transduction process where that signal is going to be changed through a series of steps that will eventually produce a substance that will cause the plant stem to respond. And for the last part of the response, the plant stem is actually going to bend toward the light. Now, notice that I didn't go into detail about how that happens with the chemicals, and it's okay. If you know more that auxin is the substance that's involved, you could certainly go ahead and be more specific about how that phototropism works, but this is about how phototropism is an example of that cell signal pathway. In part C, we're looking at treatment group 4, and we're looking at the fact that we are removing the plant tip and placing it back onto the shoot using a permeable barrier. And that hopefully everybody knows what permeable means. And then we want to look and see, explain the effect of this change on the response that's seen in treatment group 4. Treatment group 4 has a permeable barrier, which means that the substance is able to move from the tip to the stem, and so the plant bends toward the light. In part D, we're going to justify the claim that the cells located at the tip of the plant shoot detect light. And in our response, we're going to use evidence from both treatment group 1, which we've identified as the control, and treatment groups 2 and 3. So in treatment group 1, the tip is unaltered and the plant bends toward the light. In treatment group 2, the tip is removed and the plant no longer bends toward the light. In treatment group 3, the tip is covered with an opaque cap that blocks the light and the plant no longer bends toward the light. So it's something about the light and the tip that is causing that response. In part E, we're going to add a 5th treatment group, and it has the tip removed and placed back on toward the light. Just our prediction, and now we need to justify our prediction. So in treatment group 5, the impermeable barrier prevents the substance from being able to diffuse from the tip to the stem, and so prevents the response of bending toward the light. So that explains our prediction that treatment group 5 will not bend toward the light. Okay, so that is the debrief of question 2, and I hope that your response matches these scoring guidelines. Okay, so I need to give you just a couple of items for homework. So first of all, if you go to cb.org/AP2020 Get Ready, there are 5 things that you can do to get ready before your exam. Try the demo, and I gave that to you for homework last night, and I'll remind you where to find that in the next slide. Complete your exam day checklist, and I'm going to give you an image of what that would look like. Confirm your email address. Locate your e-ticket, which will be sent to you 2 days before the May 18th exam, and make sure you check in 30 minutes early. And again, make sure you know your time zone. AP Bio exam will begin on Monday, May 18th at 2 p.m. Eastern Standard Time. So Dr. Dykstra's students, she's in the East Coast, they'd be starting at 2:30. Out here in California, my students will be starting at 11 a.m. Okay, so this is what your AP
in
exam.
Minnesota, they'd be starting at 1.
Oh, see,
we're Central Time.
Central Time. So know your time zone.
Yeah,
And
know
I
your
didn't know
time
that
zone.
about Minnesota. Okay, thank you. So important. So this is what your checklist will look like. Make sure you know your 8-character AP ID. That's going to need to be on the top of all of your responses, whether you type it, whether you handwrite it, whether you upload it in a text box or you cut and paste, attach. However you do this, you're going to have your AP ID attached to your work. And you can find that on your e-ticket or at your AP at myap.collegeboard.org. So make sure you know it. Write the name of your exam. So for us, it's AP Bio. The exam date is Monday, May 18th, 2020. And I'm using the Eastern Standard Time. So there's a 2 p.m. start time for Eastern Standard Time. So that means you should check in at 1:30 p.m. So 30 minutes before your exam start time. And then tune in tomorrow, please, because we're going to do some tips, tricks, and best wishes. For those of you that have been with us since the very beginning, we're going to give you a little bit of a send-off. And the important links are here. Again, I've highlighted that AP demo site so you can practice how you're going to upload your responses. And again, if you're looking at these links, numbers 9 and 10 really do not apply to AP Bio students. I want to thank our very special guest, Professor Dykstra, for being with us. So great to see you, and I look forward to working with you on scoring these exams in June. So thank you so much.
It was a pleasure to join you and very fun to work with you again, Allie.
All right, so we'll see you soon. See you tomorrow, kids. Okay, so bye.
Bye-bye.
Bye.